Okay, so first the recap. Last week we covered polymerase chain reaction, or PCR, and what this is it's just linearized DNA copying. You get a segment of DNA, you put it in with some DNA polymerase, and that in turn elongates the DNA and makes many copies of whatever strand that you want. You can also use this for what we call air-prone PCR. You're going to be using this in the Miniproject, where you add an external reagent. It's going to be manganese, which makes the polymerase air-prone. So it will maybe delete a few nucleotides here and there, maybe add a few nucleotides, maybe substitute a few nucleotides, and in turn it will change the properties of the DNA strand that you're of interest that you're working with. In general, the cyclical nature of RNA or of polymerase chain reaction is given in 3 steps. It starts with denaturation, where your DNA strand of interest separates into 2 single strands. Then it goes to annealing, where your primers attach to each of the ends, which with one being a forward primer and one being a reverse primer. And these primers allow for the addition of more nucleotides so it can finally replicate your part of interest. And finally, you do extension, which happens at a higher temperature. And this is, where the primers get extended and you form that fully functional piece. Okay, so the Miniproject. This is where there's not going to be very many pictures. It's going to be a lot of reading, a lot of me explaining stuff. If you have any questions, contact me in the Discord, or we can set up a Zoom call if you want a more personal like discussion of what you're going to be doing. So you have 2 options when you're doing the Miniproject. You have option 1, which is creating a new chromoprotein, or you have option 2, which is just choosing a project of your own volition and doing everything yourself. Now, I'm going to assume that you're going to be choosing the option 1 because that's just easier. Well, for one, it's easier for me, and it should be easier for you guys if you're working with groups. And your goal with this chromoprotein project is to create a new chromoprotein and submit that chromoprotein to the registry. You're going to be doing this through mutagenesis. We're going to use that air-prone PCR, and hopefully you'll get a new color out of this whole procedure. The structure of the chromoprotein determines what color it is, and therefore, if you change the nucleotide sequence of that chromoprotein, you're going to end up changing the color, which is why you use mutagenesis. Okay, so the Miniproject layout is set into, I would say, 3 stages. The first stage is the planning component, where you're outlining all the steps you're going to be doing for the Miniproject, including what reactions and protocols you'll be using, and also the key ingredients used for each of those reactions. Now, I don't want you here to outline how much of each key ingredient you need, because that's very specific to each of the protocols you're going to be doing. I just want you to know what you're going to be using, not how much. And for the outline, I want you to know what the steps are, why you're doing the steps, rather than all the little specifics and intricacies of each of the steps. Don't worry about that until you actually start doing it. Next, you get to the wet lab, which is what I consider the longest component. This is when you're going into the iGEM lab itself, either individually. You're going to be with me regardless, or with your group if you decided to do this in a group, which I recommend. And you're going to do each of these protocols one by one, and they'll build off each other, of course. And after you finish one, you'll maybe debug it if it didn't work, or you'll move on to the next step if it did. And you'll keep on going through the steps until you finally finished this whole entire protocol. And this is why you really want to know what you're doing when you're planning, because if you don't know what the steps are, or if you don't know what they do, the wet lab component is going to be very difficult, and you're going to have to learn what you need during this wet lab component. The main purpose of the wet lab component is to not necessarily learn what each of the protocols are used for or what they do. It's to learn the procedure at which you're doing the protocols. So you're learning the many steps involved with each of the protocols and how much of each component you're going to need. So, but by that fact, you're going to use the planning step to know what they're used for, and the wet lab step to know how to do it. And finally, the final segment is the diagnosing and submitting step. Here, you're going to be either diagnosing what went wrong with your mini project and trying to figure out what ways to do better in the future, even though you won't be doing it in the future unless you really want to. And if you're successful, you're going to be submitting your part to the registry, which has a few smaller steps involved with that. We'll worry about those when you get to that step, however. You want to focus on these main first two to begin with. And yeah, a big note here is I am allowing group work next semester or even this semester on the outlines. I believe that learning this stuff is much easier when you're working with other people. It's kind of overwhelming when you're working on it by yourself, which is what I had to do. I don't want to put you guys through that, so I highly recommend doing group work. You can have however many people you want in your group. In fact, the more people, it makes it easier for me because I don't have to schedule multiple days for multiple groups. Even if it's a group of like 10 people. If we have 10 people, new lab members who want to do the mini project, who are ready to do the mini project, I will allow that. I'll have each of you work on a certain component, and we can all plan this out together. Okay, so planning. It's, I would say, the most important step because you're finally you're bringing together everything you learned in all the different lab meetings I did this semester. You're bringing it together in a full project where you know what you're doing and you know what steps you're going to be doing in order to submit something at the end to the registry or to me. Here, you're going to be creating an outline of what you're going to be doing. You can do this pen and paper, or I recommend using, like Google Docs or Microsoft Word in order to create this outline. It's really up to you, though. And what I want you to include in this red box is the order of the protocols. So the protocol 1, what you're going to be doing. So say your first protocol is transformations. There's your second one's digestions, et cetera, et cetera. List out those protocols and the sequential ordering of how you're going to be doing them. Also, within each of these, I want subsections where you detail your key ingredients used in each of the protocols. You don't have to include the amounts of those key ingredients. Just say for your transformations, you're going to need chemically competent cells, and you're going to need the parts that you're going to be using to transform those chemically competent cells. Include stuff like that. Now, if you for these protocols, if you need like a certain broth, or a certain a base solution in order to do this, and don't worry about including that. Just the key ingredients that you're going to be using, the stuff that you have to determine that varies before you start each of the protocols. If you have any questions about what is a key ingredient and what is not, just contact me on the Discord. I'll let you know. And finally, I want a sentence or two detailing, you can be how specific as you want, what the protocol does. And that's so I know that you know what the protocol is used for when you start doing it in the wet lab. And for the outline, if you plan on doing the wet lab component together, I highly recommend doing the outline component together. You can contact each other in the Discord, or you can get to just use GroupMe or something, find out your groups that you're going to be working with, and work with this outline together. I would hate for you to make multiple outlines and they're differing, and it makes it all a mess when you're trying to start doing your wet lab components as a group. Okay, so I wanted to make it a little bit easier for you guys because I felt like it was a bit overwhelming if you just read the mini project, not the outline, but the thing that I gave you where it details what you're going to be doing and the general steps of what to do. I felt like it was very vague. Kent, one of the old lab managers from a few semesters or for a few years ago, made that. I don't think it does the best job at explaining what you're going to be doing. It's mainly self-taught, which is overwhelming. So what I thought I'd do is I'd make the general order of protocols that you're going to be doing. And I want you to include each of these steps within your outline and include, like I said, what the protocol does and the key ingredients. So at first, I want you to determine what you're going to be, what parts you're going to be using. Because you're making a chromoprotein, or a new chromoprotein with a different expression, so it could be like blue, pink, etc. I want you to include what chromoprotein you're going to be using, also what promoter you're going to be using in order to express that chromoprotein. Because if you just had the chromoprotein in the plasmid, then it's not going to be expressed. It's just going to be sitting there and doing nothing. And also your backbone. Now, in this, I included links to each of the recommended parts that I at least recommend. It's what I used for my mini project. The promoter is that I included with this link is a constitutive promoter, meaning in order to activate it, you do not have to add anything to the mixture. It's just active always. So it's always transcribing the chromoprotein of interest, meaning if it's transformed properly, your bacteria is going to immediately immediately create your chromoprotein without having to add any external reagent to activate it in the mixture. Also, your backbone, which I already chose for you guys. Your backbone is going to be the PSB1K3 rather than the C3 backbone. This is not the standard backbone. However, in order to make this work properly, we need a backbone with kanamycin, the antibiotic resistance gene. Notice that each of these parts, this has ampicillin resistance, this has chloramphenicol resistance, and this has kanamycin resistance. It's good that each of these are different, because if they weren't, you wouldn't be able to do 3A assembly, 3 antibiotic assembly efficiently, or just to begin with. That assembly protocol requires you to have different antibiotic resistance genes in each of your parts in order to properly screen them later. So I recommend when you're creating your outline, I recommend you use these parts that I've linked. Click on these links and read the pages, know what they are, know what they do. I recommend using them. This promoter, this chromoprotein is the MLCP blue chromoprotein. So if you didn't mutate this and you put it in bacteria with a promoter, it should make the bacteria blue. Your goal though is to not have it blue. Your goal is to make different, like other colors, so like green, purple. Your next step is the transformation step. It's where you're taking these parts that you've resuspended from the kit plates and you're preparing them in order to mini prep them. So you resuspend them from the kit plates, you extract them, put them in little epitubes, and then you transform each of these parts individually into different bacteria. It's going to be the same kind of bacteria, but it's going to be segmented. One tube's going to have your promoter, one tube's going to have your chromoprotein, one tube's going to have your backbone. I guess not your backbone because it's already cut, so don't worry about that. Only worry about your promoter and your chromoprotein. In order to follow this protocol correctly, I recommend either clicking on this link to go to my slides where I covered transformation, specifically chemical transformation, or you can read pages 61 through 64 in the lab manual where they detail the transformation protocol and what you're going to be needing in order to do that successfully. After you successfully transform your bacterial cells with these first two parts, you need to screen them. So you're going to plate them. And then you're going to screen them to see if they transform properly. Of course, you only want the bacterial colonies that have the plasmid of interest within them. After you successfully screen them, you begin your mini prep procedure. You can click on this link to visit my slides again where I covered mini preps, or you can read pages 50 through 53 in the lab manual where they detail mini prepping. And the goal of this mini prep step is to extract the plasmids that you just transformed in order to get higher concentrations of the plasmid of interest. Because when they're in the kit plates, they don't have very much concentration. It's like one nanogram, 1 to 100 nanograms, depending on how it was stacked in there. And you want around 400 nanograms. So the goal of mini preps is to increase that concentration, just have the plasmid by itself in a tube that you can use. Next, I want you to outline the air-prone PCR polymerase chain reaction step. This is the step that you're going to be using in order to mutate your chromoprotein. Notice that your promoter is not used in this step. It is only your chromoprotein, so your blue MLCP gene. I linked the protocol that another iGEM team used in the past. You can look through that. That gives you the description of what you like, how much of what each ingredient that you need. You can click on that if you want. But the main thing here that I want you to determine for PCR is your forward and reverse primers that you're going to be using for your chromoprotein gene in order to do the PCR extension, the annealing step. And I also want you, I recommend at least using these two primers I think this one's the forward and this one's the reverse. I could be mixed up on that. But click on these links, read about these primers. I recommend using these for your chromoprotein gene. And also, if you don't really know what primers are, what they're used for, I recommend clicking this link where it details on the iGEM page the different types of primers and what you'll be using them for your air-prone PCR. Next, I want you to outline the 3A assembly protocol. This is where you start the 3A assembly protocol. So you start with the double digestions, where you're starting to cut your parts away from the plasmids, so they're just the part themselves. It's not the external, all the external gunk that you don't need. It's just the chromoprotein. It's just the promoter. It's not the backbone. Notice that your backbone is already cut, so you will not have to do the double digestion for that step. The main thing I want you to determine here is what restriction enzymes that you're going to be using for each of the parts. So your prefix part, which is your promoter, is going to need a certain amount of restriction enzymes, and your suffix part, your chromoprotein, is going to need a certain amount and a certain type of restriction enzymes. I detailed this in the double digest section in my slides, which I linked here. The prefix part has a certain type and the suffix part has a certain type. You can find it by clicking on that link. Alternatively, you can read the pages 56 through 58 in the lab manual, where it goes over double digestion and restriction digestion protocols in general. After you cut these parts and they're linearized, your two parts being your unmodified promoter gene and your now mutated chromoprotein gene, you're going to want to run these mixtures of cut parts in a gel electrophoresis in order to remove the parts of interest from all the external stuff that you added to the tube. So when you cut these, you cut it with a variety of things. So you have your part, you have your fully functional plasmid part that's not cut yet. You have your restriction enzymes, and you have your base solution. Now, when you're going to ligate these together in the future, you don't want all that external backbone in there, and you don't want your base solution in there. You want it separated. You want your parts separated from all that stuff. And in order to do that, you need to do gel electrophoresis, which I linked to my slide presentation right here. And what you do is you use this to purify your parts of interest, your linearized parts of interest, into just in order to use them successfully in the ligation step. You can also read pages 67 through 72, specifically the gel extraction protocol, which I didn't go into very much detail with. I highly recommend reading a lot about the double digest step and the gel electrophoresis step, though, because this is where most people mess up. After you've segmented your linearized parts, so your promoter and your mutated chromoprotein, you now want to ligate and assemble them together into a fully functional composite part. Notice you finally have brought back your backbone that was already cut, because you're going to be putting your promoter, your mutated chromoprotein, and your backbone all together into a circular plasmid. You can click this link for the ligation assembly protocol that I included on my slides. And also, if you want to learn about 3A assembly from the iGEM website itself, including how much of what that you need and the general procedure of what you follow this link, and that will take you to the iGEM page for 3A assembly. After you make your composite part through this 3A assembly protocol, you're finally going to transform bacteria again. So notice you've made kind of a loop. You started with transformations, and you're ending with transformations. But instead of transforming the parts individually this time, you now have all the parts connected. They're all one part. They're a composite part. So you're going to transform the bacteria with this ligated composite part in order to diagnose if your ligation and restriction digests were successful. You can click that link again. It will take you back to the relinked transformation slides, and also 61 through 64 for this protocol. The goal here is to have your composite part grow on plates. So if it transforms properly, you'll have a plate, and you'll have some of the mutated chromoprotein that has like blue, some will be pink, some will be purple, because the mutations are random, right? They're stochastic. So it's going to be random colors in random positions on your plate, and you pull off colonies of each different color in order to diagnose them later on. We'll worry about that towards the end, though. I want you to mainly focus on these beginning few steps first. This is where you start beginning to screen and sequence, and you prepare your and submit your part for the registry. It's a bit ahead of what we're doing right now, so focus on the earlier stuff. Okay, so after you either successfully conclude your mini project, or you reach a part of it where you can't complete it or you get stuck, you're going to have to submit that to the registry. Here I outline my plan, at least for the outline of the planning submission for the outline you guys are going to be creating. I restated again, in your outlines, I only want you to focus on the protocol that you're going to be doing, your key ingredients, and also the order of the protocols that you're going to be doing. I also, I can't stress this enough, I do not want you to worry about your ingredient amounts until you actually begin working on each step individually. If you worried about the ingredients amounts, it would be it'd be too specific, your outline would be massive, because you'd have to include the steps at which you'd be doing stuff, and most of the procedures, actually you have to complete the previous step in order to even know how much of what you're going to be needing for the next step is. So yeah, don't worry about the ingredient amounts, worry about the protocol, the purpose of what you're doing, the key ingredients, and the order of the protocols that you're going to be doing. I included my example outline in this link if you want to view it. However, just know that it's not a very good one. I was way too specific and it was way too long. I want you to only include this. I made the mistake where I included like every step. Don't do that. Don't focus too much on the little things. Focus on the concepts and the purpose. The goal is to understand what you're doing, rather than focus on all the little intricacies. Make sure to ask me questions here. I've already gone through this so I can help you, especially if you're confused on what something does. But generally, use the outline use my outline as your general outline of how you're going to make your outline. So if you have any questions of how you want to structure it, ask me. In general, though, you're going to have your purpose at the top, so what you're doing, and then you're going to have each of the procedures sequentially after that. So each of these steps with your purpose, ingredients, and order, and the order, right? Because you included the order. Okay, next I want to include the wet lab component, because this was all just the pre-wet lab. Next semester, we're going to be doing each of these steps in the project one by one. It's during this point that you're going to focus on each of the steps ingredients and the inner workings of how to conduct the steps individually. But you're only going to do this as you do them. So don't worry about the step afterwards. Worry about the step you're on, and then worry about the ingredients for each of the steps you're on. When you're doing wet lab, I'm going to have each of the individuals include and make their own lab notebook. The lab notebook is different from your group. It's going to be an individual basis. That's so you guys can each get an understanding of how to fill out a lab notebook, because I don't want just one person doing it for the group. And during the lab notebook, I included a link to my lab notebook over the past few years. It's pretty long. Just scroll through it, get a general gist of how it's structured. Alternatively, you can look at the lab manual pages 27 through 29, where it gives a description of what should be included and what shouldn't be included. And the main idea for each lab notebook is before you come to lab, you're going to be filling out like a little pre-lab notebook thing where you outline what you're going to be doing that day, so you don't have to worry about it when you get to lab, and we can get out of there pretty quick. And also after you finish the lab, You're going to detail what you did during each meetup, your products, your results, etc., your notes. All of that is going to be included in the lab manual, or yeah, the lab. I should say lab notebook. I recommend, my last recommendation for this is using the created outline that you're working on over the break as a conceptual basis for what you're going to be doing, and then using your lab notebook as more as the specific outline of what you're going to be doing, where you're going to be adding ingredients and adding specific information. I included the general outline of what the lab manual lab notebook looks like. It starts with the date, then it goes to the purpose, so what you're doing, the protocol, how you're doing it, maybe include what the protocol's called, or maybe even the steps at which you're using in the protocol. You can make it as specific or as vague as you want. Just make sure that we know what protocol you're using. Then the products, so what you made during the procedure and where they came from. Your results, you may not be able to include those results into until like a day or a few days after you do this. Just make sure to add it later on, the results. So what your concentrations look like, if stuff grew on your plates, stuff like that. And then notes if you made any mistakes or if anything went really well, any side comments that you have, stop time, so when you finished, and also I didn't include it here, but the date at which you finished. And then the next step, so what you're going to be doing next time in order to continue with this procedure, or if you're doing a new procedure next time. Okay, so with the mini project diagnosing and submissions section, I mentioned this earlier, but you don't have to fully complete your mini project. If you reach a part where you can't complete it, you get really stuck. The goal is just to try your best. It's not to actually submit a part at the end. It's to get you familiar with the procedures, get familiar with the lab notebook, how to fill it out, and finally cement the knowledge that you learned throughout all the lab committee meetings that I did throughout the semester. At the end, we'll either submit that new composite part that you made, the mutated chromoprotein composite part, or we'll diagnose what went wrong with your procedures in order to learn and better understand what to do in the future.